लिंक पर क्लिक। हेलो सर, सुन पा रहे हैं आप? गुड मॉर्निंग टीचर।
हेलो।
हाँ बोलिए सर। सर सुन पा रहे हैं आप?
यस।
हेलो यस।
यस यस
नो।
आई कैन हियर यू।
ओके बोलिए सर।
You can turn off your video and mic and before beginning this class I will give a
हम्म
brief generic
हम्म
set of instructions so that all
हम्म
participants can turn off their video and mic so that I can proceed with the class. Okay, you can interact
हाँ,
with me via the chat box. आप chat box में मुझसे बात कर सकते हैं.
ठीक है सर। ओके थैंक यू।
Yes.
थैंक यू सर। सर चश्मा पहन लेता हूँ थोड़ा। सुनो चैट बॉक्स में मुझसे आप बात करो चैट बॉक्स पे। दूसरी चैट बॉक्स दी नहीं। चैट बॉक्स दी नहीं सर। रुको रुको। चैट
Hi students
यस
and
सर।
welcome to the second week of
हम्म
environmental biotechnology. Every
हम्म
Saturday 3 p.m. to 5 p.m. We will be
हम्म
having this class where we will try to
हम्म
solve some of the exemplar questions from the corresponding week lecture. So today we'll be
हम्म
solving all the lectures that you have covered, that Professor Pinaki sir has covered in week 2
सर को
of
बोल।
this course, and general set of instructions for all the participants, that
जी
please
सर।
turn off your video and mic for smooth conduction of this session.
जी सर।
Allow me to present the presentation, as well as I will be the only one who will be talking, so that you can interact with me via the chat box. We will have a MCQ
जी सर।
interactive session. So I would ask all the participants, please turn off your video.
ओके
Yeah,
कम
please.
ऑन कम ऑन।
Please turn off your video and mics, and you can talk to me in the chat box. We will have a interactive session. This
ओके ओके।
this questionnaire that I have developed is very similar to what you will be facing in your assignments for this NPTEL course. So let's have a interactive session, and I want everyone to participate in this class and try
जी
to
सर।
answer
ओके
these questions. Yeah.
ओके
Can I ask Dheeraj to please switch off your camera? I think I can still see your video. You can switch off your camera, Dheeraj, and
ओके
your
ओके
mic as well.
ओके सर।
Yes.
कैमरा कैमरा में क्या प्रॉब्लम हो गया है? सर इस इंटरनेट की प्रॉब्लम चल रही है सर इधर थोड़ा सा।
No issues. At least you can turn off your mic so that we can start the session. Right? Can you
ओके
please do that, please?
माइक
Thank you.
क्या हो गया है?
Okay, I'll just share my screen in a minute. Please wait for a while. I'm sharing my screen. This is in progress. Can everyone see my screen? I don't think it's shared yet. Can everyone see my screen? Please let me know in the chat box. I think I think you are not able to see it.
सर कैसे दे रहा था सर इसके क्या करें
Yeah, Krish, you want to say something? You have raised your hand.
The screen is not visible, sir.
Screen is not visible. Okay, okay. Yeah.
क्या करें
Meanwhile, I'll keep admitting everyone who is trying to join this call.
चैट बॉक्स है चैट बॉक्स रुको देखता हूँ कटो क्या है हम काटते हैं
Dheeraj, please turn off your mic. I think your mic is still on. Can you do that? I don't know why I'm not able to share my screen with you.
पहली बार लाइव पर जुड़े हूँ और इसके बाद थोड़ा बहुत समझ नहीं आ पा रहा है इसलिए आई एम सॉरी स्क्रीन पर साफ तस्वीर आ रहा है कि नहीं ये जो दिखाई सर धीरज दास
Yeah, yeah, I'm trying to do that. I'm sorry for the delay. Maybe I'll stop presenting and I think this would help me share entire screen. Now maybe you can see it.
Yes, sir.
Yeah. Sorry for the delay. Thank you for waiting and again once again welcome you all to the second week of environmental biotechnology. My name is Ashutosh Vera and I'm a PMRF PhD scholar at IIT Kanpur. So I'll be the teaching assistant for this course and during these sessions on Saturdays we will be solving example questions from week 2 lectures. So okay, and we'll follow a consistent format that those who have attended my first class would already know, but I'll repeat it for all the students who have joined right now. Please turn off your mic and video chat during this whole class. You can interact with me via the chat box. The format of the presentation will be like this. I will post 10 multiple choice questions which is similar to what you will be facing in your assignments, right? And then all these questions are from within the week 2 lecture set, and we will try to I will ask you the what is the correct answer. You can let me know what is the correct answer in the chat box. You can just write option A, B, C, D. Then a minute or so after collecting all the answers, I will reveal what is the correct answer and then we will discuss briefly on that, thereby clearing your doubts and covering the topic as well. So it is a quick revision of your topics that you have read in this week, as well as a practice problem set for you guys. And this class is a recorded class, so the recordings will be available on YouTube and provided to you via NPTEL. So the recordings of this class as well as the slides will be provided to you through the link. Okay, without any further delay, let us start with the first question of this session. Just a minute. May I just turn off my video as well? Yeah, I hope you can hear me now well and clear. So this is the first question of this session. Why is the conventional definition of species not adequate for microorganisms? This is the question. What is the conventional definition of species and why does microorganism does not fit it? Option A, microorganisms have much greater genetic diversity. Option B, microorganisms reproduce asexually. Option C, microbial ribosomal structure is different. Option D, microorganisms are too small for the physiology to be studied. And option E, microorganisms evolve too quickly. Now these are multiple choice questions, so only one of these choices is the correct answer. Take your time and read the question again. I hope you can see it on my screen. And let me know in the chat box which do you think is the correct option. Let's take a minute and answer. I see many raised hands in the chat. If you are just trying to answer the question via talking to me, you can just post your option in the chat box. Okay, Krish has said option B. There are already many answers. Tapsi, option E. Olivia, E. Yashashvi. I remember these names from the last class. Yashashvi, Yashwant, all are saying E. Akansha is saying B. Okay. I think we are less in strength today. I think 9 or 10 people. In the last class, we were around 15 to 16 people. No issues. So I'll read the question again. This is also for those who will be watching the recorded lecture. Why is the conventional definition of species not adequate for microorganisms? I'll encourage everyone present in the class to at least guess the answer if you do not know it, and then I will reveal what is the correct answer to this question. Okay, I think it's time for me to tell you what is the correct answer. The correct answer to this question is, why is the conventional definition of species, why doesn't it fit microorganisms? Option B is the correct answer. Microorganisms reproduce asexually. So this is one of the reasons why that conventional definition of species does not fit microorganisms. So Krish and Akansha are both right. Well done. You have answered option B, and all those who have answered option E, better luck next time. So option B is the correct answer. Now let us understand what is the general definition of a species. The general definition of a species includes four important characteristics. Two organisms are said to be species when they have similar physical characteristics that we can observe. If we observe microbes under the microscope, then if they are having almost similar physical characteristics, one of the general definitions. Interbreed in the wild and have fertile offspring. This is one of the most important definition criteria for fitting into the general definition of species. Animals of the same species or organisms of the same species should be able to interbreed in the wild and have fertile offspring. And one species should be able to be distinguished genetically from other species. The genetic composition of one species should be different, and they should have common ancestry or evolutionary derivation. So these are the four general characteristics of a general definition of to fit into species. Now we know that microbes are well known for asexual reproduction. This is an example here, vegetative reproduction by binary fission in the bacteria, where there is a single cell in which the DNA is copied by a replication and an internal septum arises, which then divides the parent into two daughter cells individually. And the separation of these cells gives rise to two independent identical cells. So the conventional definition of species is based on interbreeding and reproductive isolation. It means there is no scope for vegetative reproduction in the conventional definition of species, which does not apply to microbes that primarily reproduce asexually. So it's also that asexual reproduction is one of the primary modes of reproduction for microbes. That is why microorganisms do not fit the conventional definition of species, making this definition inadequate for them. So I want you to look at these four definitions. General definition of species: they should have similar physical characteristics, interbreed in the wild and produce offspring, genetically distinct from other species, and should have a common ancestry. And microbes, being a species which is primarily vegetatively reproductive or asexually reproductive, does not fit into the conventional definition of species. So option B is the correct answer. Microorganisms reproduce asexually is why the conventional definition of species is not adequate. I hope everyone is with me. Everyone understood this question. If yes, just let me know in the chat box with a yes or a thumbs up. Good. Great. Thank you for the response. Quickly moving on to the next question. Question number 2. Again, so this time I have designed a little lengthy and complex questions which will compel you to think. Now, which of the following statements is false? There are 5 statements and you have to identify true and false. Which of the following statement is false? Only one of them is correct. Option A, nutrient rich ecosystems have moderate species abundance. Read the statement carefully. Nutrient rich ecosystems have moderate species abundance. Option B, all cells of the same microbial species show at least 70% DNA hybridization. Option C, members of the same microbial species have an average nucleotide identity of at least 30% at whole genome level. Option D, extreme ecosystems have low species richness but can have high species abundance. And option E, species richness is the total number of species per phylotypes present in an area. So each of these statement is a is from the each of these statement is mentioned, and you have to tell me which of these statement is false. You have to think a little bit about these statements. One of them says talks about nutrient rich ecosystems, the other talking about microbial species and DNA hybridization. Some other options talk about species richness and species abundance. Now let's take a minute and collect some answers in the chat box. So people who have joined this class, I would encourage everyone to answer, because I will be I will be regularly monitoring the chat box for your answers. And a request to everyone who have joined recently as well as the existing students, please turn off your mic and video chat while attending this class for the smooth conduction of the session. Okay, I see some answers. Saida has answered option C. Okay. Krish says option D. Yashashvi option D. And there is one more answer. Akansha says option D. Tapsi is also going with option D. Okay, what is option D? Extreme ecosystems have low species richness but can have high species abundance. Okay, let us see what is the correct answer to this question. Which of the following statement is false? I will repeat all the options again. Nutrient rich ecosystems have moderate species abundance. This is absolutely true, this is not false. All cells of the microbial species show at least 70% DNA hybridization. Now, option B and C are little confusing for the sake of MCQs. Members of the same microbial species have an average nucleotide identity of at least 30% at whole genome level. Extreme ecosystems have low species richness but can have high species abundance. And species richness is the total number of species phylotypes present in an area. And the correct answer to this second question, which is false, is not D but option C, which is members of the same microbial species have an average nucleotide identity of at least 30% at whole genome level. This is the answer which is false. I mean, option C is the correct answer. Now let us understand each option in detail. Okay, with me? Let us understand the concept of species abundance and species richness. Now, in a patch or an ecosystem, what do you mean by species abundance? The total number of individuals of a given species within a given area. In an area, the total number, the sheer count of individuals. Let's say there are 17 individuals, irrespective of different species. They can be of different species or same species. And what is species richness on the other hand? total number of species within a given area. What are the different types of species present in a given area? So let's say 17 animals, but there are 6 species comprising of this 17 individuals. So that is the basic difference between species abundance and species richness. Understood? Now let us understand what happens in extreme ecosystems. Since many people have answered option D, I'm trying to make them understand what happens in extreme ecosystems. So let us take an example. Extreme ecosystems like the Arctic Ocean have high species abundance but low species richness. This is a true statement. This cannot be a false statement. Why? Because the harsh environmental conditions limit the number of species capable of surviving. That is very true because many different types of species cannot survive in a extreme harsh developmental condition. In an extreme ecosystem like Arctic, only those species which have adaptiveness to the ecosystem can survive. So the species richness is very low. Low species richness is due to extreme cold, limited sunlight, and short growing seasons. Only highly adapted organisms. That is what I just said. And these are the examples like the penguins, polar bears, killer whales, and walruses often occupy Arctic ecosystems. And why should there be high species abundance? Now you imagine wherever there is low species richness, only the species that are adapted to the Arctic can exist in large numbers due to reduced competition and abundant resources. Now the ecosystem is abundant in resources for the very limited amount of species that exist there. So they grow in number. That is why extreme ecosystems do have low species richness and high species abundance. You have to remember it like this: extreme ecosystems, low species richness, and high species abundance. Okay? Then moving on to nutrient rich ecosystems, which was one of the first options. What happens in nutrient rich ecosystem, which is very opposite to extreme ecosystems? Again, explaining with an example, nutrient rich ecosystem is a tropical example. Tropical rainforest is the best example for a nutrient rich ecosystem. Now, nutrient rich ecosystem often have moderate abundance because of high nutrient richness, it can support a large variety of species. So there is high species richness but very moderate abundance because no single species can grow in abundance. They maintain a population size that are balanced, that are neither extremely high nor extremely low, but instead exist in intermediate numbers due to ecological dynamics. So there often comes in dynamics into picture such that the birds eat something which is not the food of any other animal. They interdepend on each other. Tropical rainforests such as the Amazon are characterized by high species richness due to abundant resources like the sunlight, water, and nutrients. However, the abundance of individual is often moderate because of competition among the same shade of resources, and rainforest species face constant pressure from competitors and predators. So you also have to remember that there are a lot of predators in the rainforest as compared to the Arctic ecosystems. This leads to partition of niches, geographical isolation, or resource utilization for different species such that they can coexist in the same ecosystem. This is why in nutrient rich ecosystems like tropical rainforest, there is moderate species abundance but high species richness. Now coming on to the correct answer, which was option C. Now talking about microbial taxonomy, in microbial taxonomy, we use a technique called DNA-DNA hybridization to assess the genetic similarity between two different bacterial strains, right? And often a value greater than 70% is used as a benchmark to say that yes, these two species are similar. If there is a value less than 70%, then these two are not of the same species. Sequences dissimilar are easy to separate species A and B, not closely related. So the 30% and the 70% was to confuse you guys. It's actually the 70% DNA hybridization level that is considered as a benchmark to add to to identify genetic similarity between two different species in microbial taxonomy. I hope it is clear to the students that why C is the correct option, and I think one of the students have answered correctly. If yes, then please let me know in the chat box. Did everyone understand, or you need me to go through it again? Very good. I got your message. Okay. Moving on to the question number 3. Question number 3 is just in front of you. Which of the following statement is incorrect? Again, true false kind of statement. Which of the following statement is incorrect? Option A: guilds are formed by organisms that are metabolically similar and utilize the same resources. Option B: all organisms living in a specific environment at a specific point of time comprise the population. Option C: microbial communities comprise of multiple guilds. Option D: only phototrophs can synthesize new organic matter from inorganic matter. And option E: allochthonous organic matter is that which enters an ecosystem from an external sources. Now there are 5 statements, option A, B, C, D, E. Which of the following statement is incorrect? Let us take some time to answer this question. Read all the statements correctly, carefully, and then let me know what is the correct answer. Salty छुपा के ही देना. You try this. I think I already see some answers in the chat box. Nandini has said option D. Akansha, Krish, Yashashvi, Olivia, everyone has answered option D. Any more answers in the chat box? Again, I would request everyone attending this class be participative. I'm not asking you to turn on your video or your mic. You can just type your answer in the chat box. Read the question. We will understand it together. It's okay if you do not know what is the correct answer. I will explain it to you. I'll make things simpler for you.
बोलो बोलो।
Yeah.
हेलो सर,
Yes.
मुझे पहले चैट बॉक्स नहीं मिला था इसलिए मैं आंसर नहीं दे पाया। आई एम सॉरी।
It's okay. You can turn off your mic and then just, if you have the chat box now, you can tell me your answer.
आप कुछ सुन पा रहे हैं? हेलो सर, मेरी बात आप सुन पा रहे हैं या समझ पा रहे हैं?
हाँ हाँ बोलिए.
ओके सर। ओके सर।
See, this is an official class. That is why I will only be instructing in English, so that anyone who watches this video later, as well as all the students who are from different backgrounds, can understand. Okay? You can, yeah,
इंग्लिश में
you can let me know what do you think in your chat box.
बड़ी
And
चैट बॉक्स में ये भी। सर, मुझे इंग्लिश थोड़ा कमजोर है।
I'm sorry, but I
बोलिए।
cannot help it. But this is an official platform, so I will only be teaching in English. Right? I'm sorry, Dheeraj. I hope you can follow the questions and the explanations. If you are not able to understand, at least you can follow the slides. आपको slides तो समझ आ रही होंगी.
हाँ हाँ जी।
Yes.
हाँ सर, मैं डीएनए के बारे में और बायोटेक्नोलॉजी अच्छी तरह से समझ आता है। आप जो भी बोल रहे हैं मुझे सब समझ आ रहा है।
Yes. Thank you. Yeah, you can mute yourselves.
यस सर।
Yeah.
यस यस।
Okay. Time to reveal what is the correct answer for this question. Which of the following statement is incorrect? And it's option D. Only phototrophs can synthesize new organic matter from inorganic matter. This is the correct answer. Well done to everyone who have answered option D. I think the whole
ठीक है,
class has answered option D.
ये बोल रहे सर। ओके।
Only this is a false statement. Now let us go through the options one by one. The first option talks about guilds. Now what is a guild? Let us look at what is the definition of a guild. In ecology, a guild refers to a group of species that utilizes same resources or exploit different resources in related ways within an ecosystem. So, a group of species which not of the same species, but they can utilize the same resources to exploit different resources in related ways in an ecosystem. So, they can thrive together. They can live together as a group of species to utilize the resources in an ecosystem. For example, a tree guild. Fruit tree guild is a perfect example for a guild. The central tree is a fruit tree, which is an apple or a plum tree, provides the main yield and structure of this guild.
आपको समझ आया?
Then comes fruit bushes and shrubs, which are planted
फ्रूट
around the trees. Example, berry plants or bushes, which offer additional harvest and fills the understory. Then comes ground cover, which are mosses and lichens and other weed and creeper plants, which cover the soil, which are low in height, and also reduces weeds and retains the moisture of the guild.
जी
Also
सर.
comes friends like insectary plants, which attract beneficial insects to support the ecosystem. There are also pollinator plants and mulch and nitrogen fixer plants, which help in providing nutrients to this fruit tree ecosystem. धीरज
हेलो
भाई,
हेलो सर,
आप
क्या
अपना
इन सब
माइक
का आंसर
आप
एक
अपना
साथ
माइक ऑफ
आंसर
कर
दे
सकते हैं.
सकते हैं? माइक ऑप्शन से बोलिए. क्या इन सब का एक बार में आंसर दे सकते हैं?
Yeah, you can. I will be posting every question one by one, and after we have gone through the explanation, we will move on to the next question. एक साथ में तो नहीं होगा. Yeah, if possible, please turn off the mic or I may have to mute you. Okay, as I was talking about the fruit tree guild, which is a good example of the guild, then there is a main tree which is providing the male yield and the structure, and there are other plants which add additional benefits to the guild. Some are nitrogen fixers, some are pollinators for the fruit plants, some are insectary plants, some plants provide ground cover, and this is an example of a guild. Now, what happens in a microbial guild? In microbial communities, the ecosystem is consisting of several guilds, such as decomposers, which break down the dead organic matter into simpler compounds, nitrogen fixers, which can convert atmospheric nitrogen into, into plants' absorbable forms, and symbionts, such as lichens, which can form mutualistic relationships. Pathogens, for a perspective of microbial community, pathogens can cause diseases, and there are also bacteria and microbes which are fermenters which can produce energy through fermentation without the use of oxygen. So all these different type of bacteria comprise of microbial communities or microbial guilds. So the first option is the correct one. Then coming on to the allochthonous
ये हमने
organic matter. So there are
लिख
two types of organic matter, allochthonous and
बोलिए.
autochthonous organic matter. And these terms
ऊर्जा.
actually refer to the sources of organic matter in an ecosystem. There can be two different sources of organic matter. Allochthonous organic matter originates outside the ecosystem, while autochthonous
कुछ.
organic matter originates within the ecosystem.
ओके.
So
हाँ
something
हाँ
from
सर
the
सर.
outside is called allochthonous, and from the inside is
आपको
autochthonous.
स्टैंड माइक आपको दिख जाता है. बोलिए.
Now the
um,
definition says that organic matter derived from sources outside immediate ecosystem is allochthonous organic matter. For example, leaf litter falling into the stream, dissolved organic matter carried by the rivers, or terrestrial organic matter entering a lake. So in a pond ecosystem or a water ecosystem, water based sea ecosystem, Leaves fallen leaves are allochthonous organic matter because they do not come from within the pond or the sea water but from outside sources. And what is the importance of these organic matter? Allochthonous inputs can be significant source of energy and nutrients for aquatic and terrestrial
ये
ecosystem, but may
अच्छा
also
फिर
introduce pollutants or alter the natural balance. So
um
these
energy
these allochthonous organic matter
that's
often impact as a source for the aquatic and terrestrial ecosystems. So that is why one of the options, I think option E was also a correct option. Now coming on to the incorrect one, which was the correct answer, which all the students answered. Not only phototrophs but also chemoautotrophs do synthesize organic matter from inorganic matter. So phototrophs convert light energy into fixation carbon, but chemoautotrophs also convert energy like fixing nitrifying bacteria, nitrogen present in the atmosphere into minerals. So they
option
also
number E.
convert
Option number E बोल
inorganic matter to organic matter.
number
And
E.
that is why the correct option was option D, which was the incorrect statement amongst all the options. That not only photoautotrophs but also chemoautotrophs synthesize organic matter from inorganic matter.
Yes. क्या करें?
I hope everyone is clear about the concept. If yes, then just let me know in the chat box, and we will
ज़्यादा
move
सही
forward.
ना होगा आंसर पूछते बचे हैं होगा आंसर अगर तो ना बजे थी ना तो आंसर तो यहाँ से दे सका इससे आंसर पूछ सकते हो सर से
Okay,
मुझे तो मैंने
great.
तो पढ़ा है ये सर पढ़ा रहा है आपसे फिर पूछेगा तुम ना
Now the
अरे यार
question 4 is on your screen.
थोड़ा
Now
बहुत
moving on
चलो जी
swiftly to question 4. Which is the correct order of metabolic distribution of microorganisms across a decreasing gradient of oxygen concentration? This is the question. So, microbes according to decreasing gradient of oxygen concentration. Decreasing means the first one would be highest oxygen concentration, and as we move to the right, there will be lowest gradient of oxygen concentration, a decreasing gradient. Option A, obligate aerobes, facultative anaerobes, microaerophiles, obligate anaerobes. Option B, obligate aerobes, microaerophiles, obligate anaerobes, facultative anaerobes. Option C, obligate anaerobes, facultative anaerobes, microaerophiles, obligate aerobes. Option D, obligate anaerobes, facultative anaerobes, obligate aerobes, microaerophiles. And option E, microaerophiles, facultative anaerobes, obligate aerobes, obligate anaerobes. Let me know in the comment box whatever you think is the correct answer to this question. Take your time, read through the options. The arrangement is from a decreasing gradient of oxygen concentration.
टाइमिंग क्या हो रहा है टाइमिंग आपसे पूछिए ये बोल रहे हैं ना डी ऑप्शन नंबर ए सी रे सी ओ सी तक तुम सी दिले
I think Dheeraj is answering something. Dheeraj, आप कौन सा आंसर बोल रहे हैं?
बोलिए सी
C.
सी
Okay.
सी
Okay. I have received your answer. I am waiting for other students to write their answers in the chat box.
राइट आंसर दिले राइट आंसर राइट
Okay. I see many answers in the chat box. Nandini has answered option A. Akansha says option B. Saida, Olivia, Krish, Yashashvi are all answering option A. Okay, Krish has answered option B. Okay. And Dheeraj has answered option C. Now let us look at what is the correct answer to this question. The correct order of metabolic distribution of microbes across a decreasing gradient of oxygen concentration. Correct answer is option A. This is the
हम्म
order of decreasing gradient of concentration. Obligate
बोलो.
aerobes, facultative anaerobes, microaerophiles, and obligate
ये बोलो.
anaerobes from highest to lowest concentration of oxygen. Now let us understand the differences between these classification of microorganisms.
ठीक है. सर ए. हम्म
So if you look at this diagram, it also gives us the definition of these classifications.
हम्म
Obligate aerobes. Try to understand this statement. Obligate aerobes. Aerobes means the ones which can utilize oxygen for their energy expenditure,
धन्यवाद.
right? They require
राइट.
oxygen and cannot survive without it. Now, obligate means it is absolutely necessary for the obligate aerobes to get oxygen. They cannot survive without oxygen. Obligate. It means ये होगा नहीं तो मर जाएगा.
हम्म.
Facultative aerobes. They use oxygen but can survive without it. Facultative
ओके.
aerobes,
ओके सर.
they can
ओके समझ
they
गया. सर
they
समझ गया.
facultative aerobes can use the oxygen but also can survive without it. So you can see across the tube there is a distribution of these microbes. Then comes aerotolerant anaerobes. They do not use
हम्म.
oxygen but can grow in its presence.
समझ गए सर.
And then comes obligate anaerobe. They are unable to use oxygen and cannot survive if it is present. So if there is oxygen, it is harmful for these species and they can only survive in absence of oxygen. And microaerophile, which use oxygen but only at very low concentration. They do not require a high concentration of oxygen. So this
ये
is the
यस सर.
order of organization from decreasing order of oxygen. I think there is a comment in the chat box. Nandini is asking, can it be first microaerophiles and then facultative? Microaerophile and then facultative. No, Nandini, I have a good
दो
argument
पॉइंट.
for you.
हाँ 2% चाहिए.
Facultative aerobes,
2 16 हो
there
रहा
might
है.
exist some
ये
facultative aerobes which actually
3
use much
40
higher
मिनट हो रहा
oxygen
है.
concentration than microaerophile.
की
So of
बैटरी
course,
से.
facultative aerobes will appear first in oxygen consumption because facultative aerobes can also survive in high oxygen, but not microaerophile. Microaerophile will need only a little amount of oxygen to survive. It uses oxygen but at very low concentration. While
रिपीट
for
ये वीडियो.
facultative aerobes, there is a range of oxygen concentration it can utilize. That is why facultative aerobes is coming before
नहीं बना
microaerophile.
बोल दो जाने से. बन हो रहा है
I hope I
सर.
cleared your doubt, Nandini.
ओके.
So
हाँ
it is
सर.
obligative aerobes.
माल.
Okay, Nandini is asking to repeat whatever I
नंदनी जी.
explained. So facultative aerobes use oxygen but can survive without it. This is the definition, right? So there can be many number of facultative aerobes. If you see this distribution, there can be some organisms which live here and others which can live at the top with highest oxygen. But these are not present in the microaerophile. Microaerophile only requires low amount of oxygen. So in increasing order, if you put them in the group of facultative aerobes, there might be some organisms which also thrive at higher oxygen concentration, higher than what a microaerophile can tolerate. That is why facultative aerobe is coming before microaerophile. Now is it clear? Yes. Cool. Very good. And here are some of the examples of the classification I just mentioned. Aerobes, microaerophiles, facultative anaerobes and anaerobes. There are many well-known pathogens across these classifications like Campylobacter jejuni, Vibrio parahemolyticus, Bacillus cereus are aerobic bacteria. E. coli is one of the favorite Microaerophiles. Salmonella species are facultative anaerobes and Clostridium are complete anaerobes. Clostridium does not survive in presence of oxygen. So the tetanus bacterium, which is Clostridium tetani, is an anaerobe. Okay, so we all learnt about the classification of microorganisms based on their oxygen usage. Is it clear to the students? Okay, Nandini is asking, is E. coli a pathogen? Yeah, there are some species of E. coli which are pathogenic to humans. So we generally culture E. coli and work with them in the lab or research settings, but those E. coli have mutations and there are other species of E. coli which are pathogenic to humans. Yes. Okay. So people are still trying to join this call. I will keep admitting anyone who is requesting to join this call. Everyone is welcome to my class, and please participate in this discussion and make the most out of it. You know, we are on the fourth question, moving on to the fifth question with lots of time on hand. Question number 5. Which of the following statements about geochemical cycles is false? Now we are moving ahead in the lecture week system. Which of these statements about geochemical cycle is false? Option A. All microorganisms can convert inorganic minerals to their bioavailable forms. Option B. Nutrient cycling involves oxidation reduction reactions. Option C. Nutrient cycling results in change in chemical speciation, solubility, mobility, or toxicity. And option D. Biogeochemical cycles of different elements are interlinked. And option E. Carbon is the only major element that can be fixed by higher eukaryotes. Here are all the options before you. Take a minute, read through the options and let me know which is the correct answer of these. All these are related to geochemical cycles. Just wait a minute, I'm moving somewhere to a quiet place so that we can discuss the answers. Meanwhile, you have the time to tell me what you think is the correct answer. I hope everyone is using this live TA session as an opportunity to revise their concepts, whatever they have read through the lectures. Please use your memory and your knowledge and try to understand these concepts, then it will be easier for you guys to remember, right? Nowadays it's very easy, uh, to use Google or AI services to just generate an answer, but let us try to understand the concept together. We will spend ample amount of time on explanation. That is why I have at least 2 or 3 slides after each question for us to understand the concept so that you do not have to visit your lectures again. Now, which of the following statements about geochemical cycles is false? Let me check the comment box. Okay. Olivia says option A, Saida option E, Tuhin option E, Akansha A, and Maushumi says option A. Okay. Mixed answers from everyone. Any more answers that I should be waiting for? I think it's time for me to tell you what is the correct answer. Which of the following statements about the geochemical cycles is false? I think Olivia changed her answer to option E. Okay. The correct answer is option A. All microorganisms can convert inorganic minerals to their bioavailable forms. Right? This is the correct answer. Option A is the correct answer. I think Olivia had given a correct answer, then she changed it. Sorry, Olivia. Akansha and Moshami were correct. Tuhin and Saeda, maybe next time in the next question we'll see whether you are correct or not. Now, what is a biogeochemical cycle? So your voice is breaking sometimes. Okay, let me let me just sit down again. Try to connect again. Maybe my internet connection is a weak. Just give me 2 minutes. Yeah, I think now you'll be able to hear me properly. Yeah, I was telling you guys about biogeochemical cycles. What is the biogeochemical cycle? A biogeochemical cycle refers to the natural pathway through which essential elements and compounds are circulated, recycled back between the living organisms and the environment, including both biological and geological processes. That is why we called it bio and geochemical cycles, in which nutrients and chemicals are recycled between individuals between the environment and everything else. Right? So here is an example of a simple biogeochemical cycle. The sunlight that is fixed by the plants in the form of photosynthesis as a producer, the plant is then ingested by the consumer, which is the animal uptake, and the plant when dies is decomposed by the decomposers present in the soil, and decomposition releases heat. Consumers burn the plant food into energy, and they also do respiration. Plants also do respiration and provide the heat, which is then recycled back. The whatever the energy in the form of sunlight and heat came to the cycle, to the environment, fixed by the producer, is now returned to the environment again. And there are rocks containing water and minerals which are also uptaken by plants and released to the environment. Now looking at the options which were incorrect ones, like the true ones, because the question was which of these options are false. Option B was nutrient cycling involves oxidation reduction reactions. This is the true statement since redox reactions are central to cycles like nitrogen, sulfur and iron cycle. So these are some of the nutrients which have their independent biogeochemical cycles, and microbes play a key role in these electron transfer processes. So all these fixation of these metals like iron or nitrogen fixation or sulfur cycle involves oxidation reduction reaction via the acceptance of electrons and donation of electrons, right? So option B is true. It is a redox reaction. So here is an example of the carbon cycle in which CnH2ON, which is the glucose molecule, can undergo oxidation to give out carbon dioxide plus water, and the carbon dioxide, the the sunlight can be used in the form of photosynthesis with oxygen, making it a oxidation reaction. Sorry, plant fix this energy to make glucose, and this glucose can be broken down into carbon dioxide and H2O again in a oxidation reaction, while reduction reaction is the photosynthesis which gives rise to the glucose. Option C, nutrient cycling results in changes in chemical speciation, solubility, mobility, or toxicity. So, this statement means that chemical speciation, different species or chemicals are formed when there is nutrient cycling. Changes in chemical speciation can alter how easily a nutrient dissolves, moves through the environment, such as the example of nitrogen fixation. Nitrogen is fixed in the form of nitrates and nitrites. So there are many species of nitrates and nitrites which exist in parts of the cycle, which helps its movement through the environment, and it also affects organisms in the nutrient cycle. For example, iron 2 plus versus iron 3 plus, or arsenic or the nitrogen, which have different toxicity and solubility with oxidation states. Now, option D and E, biogeochemical cycles of different elements are interlinked. This is also a true statement since the carbon cycle affects the nitrogen cycle and vice versa. Best example, carbon and nitrogen cycle. Microbial activity can simultaneously affect multiple elemental cycles. We know that rhizobium are the bacteria, nitrogenous fixing bacteria, which resides in the plant roots, and these activity can simultaneously affect multiple elemental cycles, not just the presence of rhizobium but the presence of mycorrhiza, the fungus, as well as carbon fixation by the plants can affect each other. Carbon is the only major element that can be fixed by higher eukaryotes. This is absolutely true. In higher eukaryotes, carbon is the only element that can be fixed. Only when we include chemoautotrophs and lower microorganisms, they can fix things like nitrogen, sulfur, and which are prokaryotes. So the only prokaryotes are able to fix nitrogen or sulfur, not the higher eukaryotes. Higher eukaryotes in this category, only producers or the plants which can fix the carbon dioxide present to glucose are known as the autotrophs. The carbon is the only major element that can be fixed, but only lower prokaryotes have more diversity in element fixation. While the correct answer, which was option A, all microorganisms can convert inorganic minerals to their bioavailable forms. Not all microorganisms, but certain specialized microbes. That was the catch in the statement. Not every microorganisms, but some, a subset of those microorganisms, some specialized microbes, like nitrogen fixing bacteria, sulfur bacteria, and iron fixing bacteria, right? All these are specialized groups of microbes, and not all microorganisms can convert these minerals. That is why option A was the correct answer. And carbon is the only major element. People got confused, but I just explained why this is not the correct answer. I hope it is clear to the students. Then we can move forward. If there's any doubt or you want to repeat, if you want me to repeat, then I can. Clear. Very good. Thanks, Akansha and Moshumi. Let us move to the next question. Next question, number 6. Again, a true false based question to test your knowledge. Which of the following statement is true? Option A, microbial communities are self organizing. Option B, microbial communities require periodic human intervention for sustenance. Option C, microbial communities have similar metabolic function and resist colonization by cells with a different metabolic function. Option D, microbial communities comprise of thousands of individuals of the same species. And option E, microbial communities do not allow alteration of their composition irrespective of environmental perturbations. So, as you can see or figure out, this is a question about microbial communities. If you remember the characteristics of a microbial community, then you will be able to answer this question. Let us take some time and collect answers in the chat box. Whatever you think is a true statement among these five statements? Okay, checking some answers in the chat box. I see many comments coming up. Nandini says A, Krish A, Yashashvi is also saying A, Akansha, Tuhin, Moshmi A, Mahima and Olivia, Saida as well. Option A. Okay, unanimously, everyone in the class is agreeing with option A. Then how can it be the wrong answer? Option A is the correct answer. You guys have answered very correctly. Microbial communities are self organizing. This is the true statement of all these options. Now, let us understand what is a microbial community. Microbial communities are groups of microorganisms that live together in a specific environment, interacting with each other and their surroundings. So, best example of a microbial community is what forms in a sewage water treatment plant or in a waste environment or any other environment, in a soil, in the wastewater, inside rivers. These consist of microbial communities, group of microbes which are living together and interacting together, changing their environment, which is making it sustainable for everyone to live. Their composition and distribution, such as the order of obligate aerobes, micro aerophiles, facultative anaerobes. We just understood the distribution of the classification of microbes based on the oxygen requirement. So, in a microbial community, there can be different types of aerobes and anaerobes which depend on factors like oxygen gradients, nutrient, and availability of the pH. So, they interact with each other and they occupy spaces in the microbial community according to their liking of oxygen gradients, nutrient availability, and the pH in which they can survive. These communities play crucial roles in ecosystem, including nutrient cycling, decomposition, and symbiosis with host. So the example that I gave of a biofilm or a sewage water plant is a nutrient cycling mechanism or a symbiotic relationship between many different bacteria and archaea and other microorganisms to form a microbial community. On my right is a example of a microbial community comprising of different bacteria all colored in different colors. There are some hydrogen producers. This is the example of a microbial community. And there are some hydrogen utilizers and fermenters. They all occupy different areas such that they can process things and fix things as a whole for the community. There is also intracellular and intercellular signaling in the communities. There are metabolic interactions and also fixation of food like conversion of sugars into hydrogen. This is a hydrogen producer. And as just as about other options, periodic human intervention is not needed for their sustenance. A microbial community on its own is easily sustainable and they often include diverse metabolic functions allowing colonization by different cell types. It doesn't mean that only one single type of microbe is a part of the whole microbial community. Many different types of microbes depending on the metabolic functions. Just now we saw that there is metabolic interactions between these different microbial communities. That is how many different cell types often occupy a microbial community. They typically comprise multiple species, not just thousands of individuals of the same species, and their composition can change with environmental perturbations. So it is very true that these microbial communities are not static. They are not stagnant. Depending on the changes in the environmental factor like pH, temperature, availability of food and resources, the microbial communities composition can change. One type of bacteria can be included into the community
and the other kind of bacteria or archaea or microbe can leave the community and can change the shape of the whole community as an output if there is a perturbation in the environment. So that is why option B, C, D, and E are all ruled out, and self assembling, self organizing microbes form microbial communities, which is the true statement. I hope the explanation is clear. This was an easy question, and I'm happy that everyone answered it correctly. Option A is the answer of the sixth question. Very good. Thanks for letting me know. We are moving ahead to the next question of this session. Question number 7. Alright, question number 7. Which one of the following methods allows us to visualize the location and abundance of specific taxa of microorganisms in an environmental sample? You have to read the question carefully. Which one of the following methods allows us to visualize the location and abundance of specific taxa of microorganisms in an environmental sample? Option A, microscopy. Option B, qPCR, quantitative polymerase chain reaction. Option C, GCMS, which is gas chromatography mass spectrometry. Option D, microarray. And option E, fluorescent in situ hybridization or FISH. Now these are some molecular biology techniques. If you remember the lecture where microbial ecology and environmental biotechnology are tightly related, and how molecular biology is being utilized to understand microbial ecology. Then Professor Pinaki sir has mentioned about all these techniques and how these can be used for specific functions. So understand the question properly. Which one of the following methods allows us to visualize the location and abundance? So what are the two things that are we doing? We are visualizing the location. We are visualizing the location and abundance of a specific type of microorganism. You need to find out a specific type of microorganism in a whole environmental sample. Let's say a soil sample or a water sample from a sewage. Will you use microscopy? Will you use qPCR? Is GCMS helpful or microarray, or is it FISH? Let me stop for a minute and collect some answers in the chat box, and for all those who will be watching the recorded video as well. And let's revisit this question in a minute where I will reveal what is the correct answer. Okay, I think it's time to check the chat box. I see a lot of answers. Tuhin, option A. Mahima saying option A. Akansha, option E. Oh, Tuhin has changed his option. Answer to option E. Saida E. Yashashvi E. Mahima has also changed her answer. Option E, and Moshami says E. Okay. Which one of the following methods allows us to visualize location and abundance of specific
taxa or type of microorganism in an environmental sample? The correct answer is option E, fluorescence in situ hybridization. This is the correct answer to this question, and everyone has answered correctly. Congratulations. Question number 7, answer option E, fluorescent in situ hybridization. Now, what is fish or fluorescent in situ hybridization? Let us understand in detail. Fluorescence in situ hybridization protocol. This is the protocol. Fluorescence in situ hybridization, as the name suggests, uses fluorescently tagged probes which can bind to the mRNA target. Now, these mRNA probes are designed specifically for sequence complementarity to their target, and they are fluorescently labeled. So, wherever our genes which are getting coded, which will transcribe from DNA to the mRNA and then mRNA to the protein, the mRNA can be detected by this probe using a fluorescence microscopy based technique. So, what is the protocol followed? You take the sample from the wastewater or the soil, permeabilize the sample, create holes or pores in the sample so as to expose the mRNA, the messenger RNAs, or the ribosome. The mRNA are usually bound to the ribosomes because they are actively translating all the time. Then you add your fluorescently labeled probe, which is fluorescently labeled probe in situ. In situ means preserving the context. Now that if you follow the question, we were actually looking for methods to identify specific taxa, a particular microbial taxa. So we know what is the target mRNA we are going to hybridize to, because which can be unique to that known organism. So we take such an mRNA, we design a complementary sequence probe which is fluorescently labeled, permeabilize the sample, hybridize the probe using buffers and other techniques, washing the excess probes to remove background fluorescence, and then visualization with epifluorescence microscopy. So it is as simple as that. You have something on your plate or on your slide which has microorganisms, and then you want to identify a particular type of microorganism and visualize and localize it. Then you use a strategy called FISH, in which you design a probe which has sequence complementarity to a gene which is specific to these bacteria, and then just put it on the slide. It will hybridize with the exposed DNA, and then you can visualize your colony. Now, this technique is often used to understand spatial organization in biofilms, like formation of microcolonies or co-aggregation or layered organization. Such a powerful technique, right? RNA fish uses fluorescently labeled oligonucleotide probes that hybridize to ribosomal RNA inside the intact cells. It can bind to the mRNA, it can bind to the ribosomal RNA. Any RNA you can design a probe against, it can bind to it. Since rRNA is abundant in metabolically active cells, it allows both for detection and indirect indication of activity. Now, it is used primarily for spatial visualization, like where in space, to visualize the location and the abundance of a particular species. RNA fish excels at showing where specific taxa are located in situ, in biofilms to see how different microbes organize and interact, and in sediments, wastewater, sludge, plant roots, and animal tissue to examine microbial colonization. In marine or soil samples to understand ecological niches. So, this is how RNA fish can be used to understand the exact location of a specific taxa in the soil or water site, in the microbial colony site. They are used for biofilms. This technique is also used to examine microbial colonization in all these different kind of samples. So I guess we now know the protocol and the principle behind the working of RNA fluorescent in situ hybridization. And I'm happy that everyone has answered correctly. Very good. Good. Is it clear to the students? If you want me to repeat any part, then I can do it. I just want everyone to be on the same page, clear about their concepts and answering everything correctly. Very good. Moving on to the next question, since we are moving very fast. Question number 8. Three more questions, including this 8, 9, and 10, and we will be done with the class. Right? Dash is a cultivation independent approach for study of microbial communities. Can be a tough question. Which of these is a cultivation independent approach for the study of microbial communities? Option A, isolation of pure culture. Option B, MPN or most probable number. Option C, metagenomics. Option D, crowded plate technique. And option E, enrichment culture. You have to find out which is a cultivation independent approach for studying microbial communities, where you don't have to cultivate anything. Isolation of pure culture, is it most probable number? Metagenomics, crowded plate technique, or enrichment culture? Let us take a minute or two to wait for everyone to answer, as well as all those who are watching this video. And then I'll come back to this question and reveal what is the correct answer. Okay, I think it's time for me to reveal what is the correct answer. Let me just check the comment box. My god, there are already so many answers. Nandini says confused between option C or D. Akansa option C. Olivia Tuhin option C. Moshumi Yashashvi option C. Mahima and Saida have also answered option C. What is the correct answer to question number 8? A cultivation independent approach for the study of microbial communities. Answer is option C, metagenomics. Again, everyone has answered correctly. Option C, metagenomics is the correct answer. Now, what is metagenomics and how is it different from rest of these techniques? Metagenomics is a cultivation independent approach that involves sequencing DNA of all microorganisms in a sample directly from the environment. So as straightforward as that. You take a sample from the environment, let's say soil, 1 gram of soil containing 10 to the power 9 microbial cells, lyse the cells, isolate the DNA from the soil sample, and then just put these DNA for sequencing to understand what are the different kinds of DNA that you are getting from the small soil sample. It allows study of microbial communities' genetic diversity and function without the need to culture them, making it ideal for complex community analysis. So in the real world or in the real time scenario, there is always a microbial community in a small, everywhere in every ecosystem and in small places. There are always microbial communities. Not a single microbe does not exist alone. So metagenomics, in which you just bulk dissociate the sample, no need to cultivate it, and directly isolate the DNA from these samples and sequence them to understand what is the genetic diversity or genetic makeup of this small sample, in which you do not have to cultivate it. This approach is known as metagenomics. And how is it different from every other method that we saw in the options? Most probable number is a statistical method that estimates microbial concentration by serial dilutions. You need to do serial dilution, in which culture the bacteria and then make serial dilutions of the tubes. Now culture the bacteria again until you are seeing loss of the count in bacteria, and multiplication by the dilution factor gives us the probable estimate of microbial concentration. Isolation of pure culture also involves growing single microbial species in isolation in the Luria botany media as we do it in the lab. It is also a cultivation dependent approach requiring microbe to grow under specific conditions and thus not suitable for studying full diversity of microbial communities, because such communities do not demand specific conditions, and thus not suitable for studying the full diversity of a microbial community. So for diversity studies, metagenomics is the best approach. Now, what is crowded plate technique and enrichment culture? Crowded plate technique also involves plating high density of microbes to observe interaction or isolate specific colonies. It is a cultivation dependent technique as it requires microbes to grow on plate. So in crowded plate technique, we use antibiotic producing microbes, or microbes which are indicator microbes, which can help us identify where like identify the microbial concentration in a sample. And enrichment culture, the name itself suggests that you have to culture the bacteria for selectively growing of bacteria or any microbe via providing specific nutrients or conditions. It is again a cultivation dependent technique and relies on the growth of targeted microbes, does not capture the full unculturable diversity of microbial communities. So, to truly study microbial communities in situ, we need to use the metagenomics approach, in which lyse all the cells, isolate the DNA, sequence them all, now identify what all these sequences belong to, which group do they belong to, and what is the abundance of certain DNAs, emphasizing the whole microbial concentration and distribution in the colony. That is why metagenomics was the correct answer, which everyone gave it very nicely, very good. Metagenomics is a cultivation independent approach and a nice technique to study microbial communities. I hope it is clear to the students. Just let me know in the chat box and we can move ahead. This sequencing will take a lot of time, Nandini asks. Yes, it should have. Back, if you go back 10 to 15 years, it would have, but now we have various approaches and high throughput sequencing methods in which we can carry out metagenomics. Understanding the total community interaction within a small species, within a small sample or a small species diversity. Okay, moving on to the next question, question number 9. 16S rRNA gene is sequenced to obtain phylogenetic identity of microorganisms present in an environmental sample. It is obtained by extracting dash of the sample. Understood the question? 16S rRNA
gene is sequenced to obtain phylogenetic identity of microorganisms present in an environmental sample. It is obtained by extracting DAS of the sample. What do you need to isolate or extract to sequence this gene? 16S rRNA. Okay, let us take a minute and collect some answers. You can take your time, think about it. What is 16S rRNA gene, which is used to sequence and maintain phylogenetic identity of microbes present in an environmental sample? This is also something that Professor Pinaki has discussed. You just need to recall. Okay, I see many answers already. Nandini says E, Akansha option A, Maushumi option A, Tuhin says B, Saida A, Yashashvi A, Mahima is saying B, Tuhin again has changed his option A, Mahima also changed her option, okay, and Olivia is saying B. I knew it that people will get a little confused. This question is a tricky one. Let us see what is the correct answer to this question. Question number 9. 16S rRNA gene is sequenced to obtain phylogenetic identity of microorganism present in an environmental sample. It is obtained by extracting total DNA of the sample. Once you isolate the total DNA of the sample, only then you will be able to sequence the 16S rRNA gene. And yes, Tuhin has mentioned the reason correctly. It is a gene. 16S rRNA is a gene, so you need to isolate the DNA to sequence the gene. And that is how phylogenetic identity is maintained. Right? We just in the previous question talked about how metagenomics is a technique which uses gene sequencing or DNA sequencing to identify different kinds of microorganisms. Now 16S rRNA is one such gene which is frequently sequenced to identify the organism phylogenetic identity. Very correct, Tuhin. Let us understand what is this gene which is 16S rRNA. Now if you look at the ribosomal subunit in the prokaryotes or in any microbe, it comprises of two subunits. The large subunit or LSU, which is 50S, where the S stands for the sedimentation constant. 50S large subunit, and there are two types of ribosomal RNA, rRNA, 5S rRNA and 23S rRNA, which make up the 50S large subunit. And the small subunit, 30S small subunit, made up of 16S rRNA. Right? Three different kinds of rRNA come together to make two different subunits which assemble together to form the ribosome. Now 16S rRNA comes from the 16S rRNA gene in the bacterial genome. Now the 16S region has many conserved regions as well as variable regions, which is mentioned in the region here. V1, V2, V3 are all variable regions, and the regions in between them are the conserved regions. Now, looking at a gene sequencing or a PCR forward reverse sequencing primer, the conservation, looking at the conserved regions and the variable regions, we can identify the phylogenetic relationship of this particular bacteria or the genome with its ancestry. The 16S rRNA gene used for phylogenetic identification is part of the ribosomal RNA genes found in total DNA extracted from an environmental sample. So you need to sequence this gene. It can be confusing, but it is not rRNA which you need to extract. You need to extract the whole DNA of the sample and then use sequencing against this gene to find out. The 16S rRNA gene is a specific part of the ribosomal RNA found in the ribosome of prokaryotic cells. Ribosomes are essential for protein synthesis, and 16S rRNA plays a crucial role in the process by forming a part of the small subunit. And the gene is highly conserved across different prokaryotic species. It has similar sequences in most bacteria and archaea. That is why this gene group is referred to as a sequencing and identifying marker for the lineage. However, it also contains variable regions that differ enough between species to be used for identification and classification. Such a convenient tool, isn't it? So, to access this tool, you need to first isolate the DNA, run a sequencing of 16S rRNA gene locus, and then understand what are the similarity between conserved regions across different species and the variable regions present in between the conserved regions. Phylogenetic identity refers to the evolutionary relationship between organisms in a phylogenetic tree, and 16S rRNA is a valuable tool for constructing phylogenetic trees determining the evolutionary relationship of prokaryotes. By comparing the 16S rRNA gene sequences of different microbes, scientists can infer how closely they are related and how they have evolved over time. So that is one information which is very crucial coming from the sequencing of 16S rRNA gene. Now, being a gene, it should be present on the DNA. So you need to isolate total DNA from a sample or from a cell, and then go for 16S rRNA gene sequencing. I hope it is clear to the students, and the next question will be the last one. If yes, then we can move to the next question. Option A was the correct answer, so many of us have answered option A. Very well done, congratulations. Nandini has a question. How about mRNA? It cannot be enough. So as we know that mRNA codes for the working proteins or the enzymes or the genes, right? So many mRNA appear to be conserved, but not many species can be distinct mRNA signatures, because many of these bacteria have same kind of enzymes and proteins. Others may have different, but the 16S rRNA is a valuable region in which interspecies discrimination can be made on the basis of these conserved and variable regions. That is why mRNA is not enough. We need the total DNA to sequence this locus primarily. mRNA will tell us what is the uh, phylogenetic. mRNA will tell us the phenotypic identity of the community. Whatever community you have purified the DNA from, if you test the mRNA signatures, then it will actually, whatever the microbes are able to convert the enzymes and the function of the community will be determined by the mRNA. For species identity, you need a more conserved region like the 16S rRNA, which also has some parts of variability. For actual whatever was mentioned in the lecture, if I remember the term correctly, the phenotypic identity. For phenotypic identity, we need mRNA signatures. For phylogenetic identity, we need rRNA signatures, rRNA gene signatures. Okay, I hope it is clear. If you have any doubts, you can ask me in the chat box. I will try to clear it, clarify it. If I'm not sure about anything, I will take up the question in the next class, read myself up, educate myself as well as you guys. Okay?
Moving on to the next question, the last question of this session. Again, a true false or a correct incorrect type statement. This is about biogeochemical reactions. Many biogeochemical reactions carried out under anoxic conditions. Read the statement correctly. Which one of the following is not a suitable explanation for this trend? Understood? Anoxic conditions, biogeochemical reactions, and which of these statements does not suit the explanation? Option A, biogeochemical cycling involves redox reactions. Directionality of these reactions are changed by ambient oxidation reduction potential. Then option B, biogeochemical cycling reactions are harmed by light exposure and only occur in aphotic environments, which were usually also anoxic. Option C, biogeochemical cycling involves reactions catalyzed by metalloenzymes which are susceptible to oxidation, making them non-functional. Option D, biological reduction of elements other than oxygen tend to yield less energy, hence most organisms prefer to utilize oxygen wherever it is available. And option E, biogeochemical cycling involves ancient microorganisms that evolved before the great oxidation event that are naturally susceptible to oxidative stress. Which of the following is not a suitable explanation for many biogeochemical reactions being carried out under anoxic conditions? Read all the statements carefully and let me know what you think is the correct answer. This is a question about biogeochemical cycles and reactions being carried out under anoxic conditions. Okay, I think it's time for me to check the comment box. Akansha has answered option B, Moshumi option B, Nandini Yashashvi B again. Okay, Tuhin and Saida think it's E, and I don't think we have many people left in the class today. Very thin population. It's okay. If people are shying about, that's okay. This is the last question of this session. You will get chance in the next session, of course. Let me reveal what is the correct answer to this question. Many biogeochemical reactions carried out under anoxic conditions. Which one of the following is not a suitable explanation for this trend? And the correct answer is option B. Biogeochemical cycling reactions are harmed by light exposure and only occur in aphotic environments, which are usually also anoxic. This is not a suitable explanation for this trend. So correct answer is B. Congratulations to everyone who has answered B. Akansha, Moshmi, Nandini and Yashasvi. Tuhin and Saida, hard luck next time. Let us understand the biogeochemical cycling and its connection to this question. Looking at each option in detail. Biogeochemical cycling involves redox reaction, of course, and the directionality might be changed by ambient oxidation and reduction potential. This is a suitable explanation because, for example, carbon and nitrogen biochemical cycle involve redox reaction, and the direction can shift based on the environmental oxidation potential, often favoring anoxic conditions where oxygen is scarce, such as in higher atmospheric conditions. When there is lightning, there is nitrogen fixation, where there is scarce oxygen, often favoring anoxic conditions in a biogeochemical cycling. Now, in in the soil where there is rhizobium bacteria fixing the nitrogen, it's an anaerobic reaction, anoxic conditions.
and the flow of the oxidation reduction reactions can change if there is a change in ambient oxidation reduction potential whenever the oxygen is scarce. Option B is the correct answer because many biogeochemical processes like sulfate reduction occur in the dark, such as deep sediments which are often anoxic, and light can inhibit certain microbial activities. But okay, I think I might have included a wrong explanation maybe. Option B was the correct answer, but let me get back to you. Biogeochemical cycling reactions are harmed by light exposure and only occur in aphotic environments. So most of the carbon fixation happens in the photic zone. Hmm. I need to look back at this question. Looking at the other options, I am confused between option B and D. Right? Here I've written that option D might be the correct answer because while oxygen based respirations yield more energy, many organisms thrive in anoxic conditions using alternative electron acceptors. And the preference for oxygen does not explain the prevalence of anoxic biochemical reactions. This seems correct to me actually because there can be other electron acceptors like sulfate and nitrate, not necessarily oxygen. It's true that oxygen reduction of other elements other than the oxygen yields less energy, but most organisms prefer to utilize oxygen whenever it is available. I think I might have to look at this question again. Uh, Maybe we will discuss this question in the next class. I have to read up myself a little bit. I'm confused between option B and D. Option B might also be correct, but the explanation I have written here, it says that oxygen is not the only electron acceptor, like we also see in sulfur and nitrifying bacteria, but definitely not option A and option C and E because metalloenzymes, which is the option C if you remember, metalloenzymes susceptible to oxidation. That is a true statement because many metalloenzymes are inactivated by oxidation, so option C is not the answer. And option E, the great oxidation event, early ancestors, early microbes evolved before the great oxidation event. That is why they are susceptible to naturally occurring oxidative stress. So this is also a correct statement. I'm just confused between option B and C because actually many anoxic reactions happen in the aphotic environments. Aphotic, which is devoid of the light, and many of the oxic reactions or the oxic zone is also referred to as a photic zone. This is the same excerpt from the slide that Professor Pinaki was presenting in his lectures, that the photic zone almost coincides with the oxic zone where there is which is the fuel for heterotrophic organisms. There is carbon fixation in the oxic zone, and all the biogeochemical reactions happen naturally in the anoxic zone, like ones made by the fermentative bacteria, nitrifying and sulfate bacteria. Hmm. This question was a bit tough, and I think even I got confused. Many biogeochemical processes like sulfate reduction occur in dark, such as deep sediments which are often anoxic, and light can inhibit certain microbial activities, which is also true. So this can be a suitable explanation. Uh, Maybe I look at it again and put this question up in the next class. So with this, we have come to the end of this session. Let me just end this session. Please remind me to take this question up in the next class. Maybe option B or D can be the correct answer. Let me just stop my presentation here and yeah. So we have come to the end of this session.
We have discussed 10 questions corresponding to lecture week 2, and we will deal with the last questions in the next class. Please read through your lectures, follow your NPTEL classes from Professor Pinaki, and read through the lecture week 3 sessions. And we'll have the next session somewhere around Friday or Saturday, because next Saturday I will be traveling, so I might not have the class. I would request the NPTEL authorities to organize it on a Friday or a Thursday or a Sunday so that I can take it if you guys are free. And this is a recorded lecture. As soon as the class ends, I will upload this recording on YouTube, and this will be available to you guys through the NPTEL authorities. Thank you for joining this class. I hope you liked it. If you have any suggestions for the presentations for the conduction of this class, you can let me know. Otherwise, I'll be ending this call. Thank you for joining. Okay, there is a help from Moshumi in the chat box. Option D is a valid explanation because aerobic respiration generally yields more energy than fermentation. Organisms will preferentially use oxygen when it's available. This implies that if oxygen is not available, alternative electron acceptors are used, leading to different biogeochemical reactions. Yes, because there are examples of sulfur fixing bacteria and nitrogen fixing bacteria, which can use an iron can also act as an electron acceptor instead of oxygen. I have been through some examples. Other anaerobic bacteria can use nitrate, sulfate, iron, manganese. Yes, yes, you are absolutely right. But I need to revisit this question because I had myself marked option B as the right answer, but then in the explanation I have written something else. So that is why it was conflicting a bit. Thank you for your help, Moshumi. I will really revisit this question. Okay. Thank you for joining. I'll end this call, and let us meet next Friday or Sunday, because next Saturday I'll be traveling along. So bye bye. Enjoy your weekend.